Hi, my name is Treefa Shwani. I'm at the University of Buffalo and I work very closely with Dr. Margaret DeAngelis.
Please tell us about the poster you're presenting here at ARVO.
So tomorrow I'll be presenting on microRNAs as a possible therapeutic target in AMD or as a likely biomarker that can be used to diagnose AMD at an earlier stage. So with this particular study, we collected donor eye tissue from a Caucasian population. This was throughout early stage AMD, in late stage geographic atrophy, so this was only focusing on dry AMD. And from this, we were able to see that based on our previous publication, which was in Cell Genomics, that microRNAs interacted with our differentially methylated positions. So this study is the first of its kind to profile microRNAs in this manner, where we do an agnostic genome-wide sequencing approach. And this has all been possible due to the Macular Degeneration Foundation and the funding that's been provided, because a lot of times it's really outside the box thinking that leads to therapeutics, and some of the projects that we're doing are considered to be higher risk by other funding agencies.
Are you only looking at RNA targets in the late stage AMD?
We also did early stage in this study, and in this case, by early stage, I mean AREDS 3 using a modified AREDS grading scale. So in this case, we referred to it as intermediate AMD. So we were looking at both early and late stage for dry AMD, and we found microRNA signatures that were unique to each of these subtypes and overlap between the two, possibly leading to disease progression. We're trying to look upstream of these messenger RNAs, so maybe I can explain that a little bit better. MicroRNAs are these small 20 to 22 nucleotide base pair structures, and they bind to messenger RNA, so then they can then repress or increase messenger RNA production. So in the Cells paper, we found that in the network of validated genes that we eventually came to, a lot of the interconnecting nodes were microRNAs. So we believe that there is a system of microRNAs that are being degraded, which is then leading to AMD disease development.
Where are you getting your tissue samples, and how can you be sure the tissue degeneration is from disease and not post-mortem degeneration?
So for what I'm presenting tomorrow, we are focusing on donors that are 60 to, I believe, 97 years of age. These are of Caucasian descent, and we are taking the macula, because this is the disease-affected tissue. And from the macula specifically, we're focusing on the macular RPE choroid, because we are comparing this to our genome-wide methylation data set, which is macular RPE choroid. And that is in the Cell Genomics paper, where we did a study looking at macula and periphery and found genome-wide methylation to be significant in the macula. So how these eyes are phenotyped, in order to increase the translational value of our work, we phenotype them as if the person is in the clinic. And to do that, we use multimodal imaging. We collect a lot of detailed epidemiological information. So from time of death to when that tissue ends up for research use, we keep that under 6 hours. So there are other labs that utilize tissue that is older, and we believe that all approaches, eventually they all benefit the patient. So multiple approaches are certainly welcome, but in our case, we wanted to minimize what you see after post-mortem time of death. You begin to see a lot of these cell death markers and pathways, and we want to find out what's truly underpinning AMD.
How are you targeting these RNA pathways, and how is that approach going to translate into a possible treatment for AMD?
One of the functional things we've kind of been moving forward with that I actually just did before I came to ARVO. So we designed these oligonucleotides in partnership with a company, Biotech, in where you can transfect cells without having to go through the transfection step. So we have cell lines from patients, and we cultured these cells, and we're utilizing them currently to look at knockdown of various things and see how that impacts protein, mRNA, and so on and so forth.
The blood you're looking at, are those current patients or are they samples from deceased donors?
Oh, these are patients that are deceased. So we took these are lymphoblastoid cell lines. So we also have the eye tissue from those same patients. So they were consented before time of death. So we also have blood, serum, plasma, eye tissue in order to encapsulate a whole picture for that particular patient.
Is there anything you'd like to close with?
I would just like to close with thanking all of our donors for the eye tissue that we're using. None of this work would be possible, and also to thank the Macular Degeneration Foundation for their funding and allowing us to kind of work on some of these higher risk projects.
It has been our profound honor and privilege to be a part of this journey. Thank you for updating us on your progress.